Okay. All right, so let's get started with the immune system. And we're going to go through all of it today. So like I said, we're just going to focus on the stuff in the immune system that's important for microbiology and what's important for us to know relevant to mainly bacteria. Very briefly, a little bit with viruses, but for the most part, we're talking about how do we fight a bacterial infection. How many of you have taken immunology already? Okay, this is going to be like, wow, that's it. That's all you're going over. Immunology has no bounds. Like that class is rough and you learn a lot. And even in immunology, the professor's probably like, this isn't even all of it. We're not getting that deep. We're staying a little more surface level here, which is kind of nice. All right. So we are discussing the kind of the opposite side of microbiology. We've been looking at the cells themselves that cause these diseases. Now we're going to look at how organisms fight them off. Particularly, we're looking at multicellular organisms that have a body system that is designed to fight off foreign invaders. So we're looking at integrated organs, tissue, cells, and cell products that all work together to stop a foreign invader from harming us. So now we're not talking about a cell, we're talking about an organism as a whole and the parts of that organism. So the main thing that the immune system has to be able to do is say, this thing belongs to me, that thing does not belong to me. And then mobilize a response if that thing that is non-self is harmful. And so it has to be broad enough to respond to anything that can be out there, whether we've seen it before or not. So the immune system is fascinating when you look at the genetic side of it, because all of these receptors that we have that are responding to different antigens or different foreign material name tags out there, all of our receptors for those arose completely by random mutation. And we basically have a receptor for anything and everything out there because we let that area mutate so much. So we have to be broad enough, but we also have to be able to recognize specific invaders that have attacked us before. So it's a really fascinating, very complex body system. And we divide it into two parts. The non-adaptive, I've never heard someone call it that one. It's the innate immune system, and then the adaptive immune system. So if we just look at those words, innate means inborn. It's something you come pre-programmed with that's just going to, the base programming that's already going to run as soon as you turn on the machine. Adaptive, this is the one that learns and is going to improve and change over time based on the encounters that it has. So with the innate immunity, we're mainly looking at barriers. The goal of the innate immune system is to say, halt, don't even come in. But if you do come in, we're going to have kind of a non-specific response. We're going to have guards that are going to be able to take out whoever just doesn't seem to belong. And this one we have to have at birth, so that way we at least have some level of protection. Now with adaptive, we're responding to specific antigens. This is a word that's going to come up a lot. An antigen, we think of it as really a name tag. So all cells have proteins or carbohydrates on their surface that act as name tags for other cells to be able to recognize. So this can be chemical, a compound, structure, anything like that that is foreign to the body. Now, technically, our cells have antigens as well. We have these name tag surface proteins and surface carbohydrates, but we don't tend to call those antigens because we mainly use antigen to refer to something that's non-self. For self, it's just our surface proteins. And all of these things are going to elicit an immune response. So it's something that the immune system recognizes and says, that's not me, let's do something about it. And the body is only going to react when it's exposed to these antigens. So it's not going to be constantly trying to find them and fight them. It only says, hey, I found this, now let's go respond to it. And then it will remember. So if you've had a disease once, it can remember it for the future and fight it off faster and better in the future. And then your second response is always going to be a faster response. The first time you're exposed to a disease, it usually takes somewhere around 14 days for your body to develop the whole response to it, for your body to create all the antibodies that it needs, all of that. But the second time, you can start taking things out in a matter of days. All righty. So let's get into the innate immune system first. There's so much with the innate immune system. There's things like complement and all that we're not even going to touch. They technically are important, but we're just going to hit on ones that will help you more with that disease project and with the specific diseases that you might come across if you become a microbiologist in a clinical setting. So, main thing with the innate immune system, remember, was it had physical barriers. So the very first physical barrier is the skin. And the skin is a very, very, very good physical barrier. It is virtually impenetrable to invaders in its intact form. If you get a cut or something like that, then they can get in. But when the skin is doing its job and it's whole, it's very hard for stuff to penetrate because we have a lot of keratin. Most of your skin is just layers and layers and layers of dead cells stuffed full of keratin, and keratin is very hard for bacteria to get through. It's also not a good source of a lot of energy for things. We produce oil called sebum, and you might think, oh, well, oil, fats, something's going to love to eat that. Yes, but it lowers the pH of our skin. So if you can't tolerate low pH, you won't survive on the skin. So you have to be able to live in a very dry environment. We don't have a lot of water because these are all dead cells. We have a low pH and we produce a lot of salt. Those are all really strong barriers against bacteria because bacteria just don't like those kinds of environments. It's hard for them to survive long term. So any type of bacteria that does colonize us and live on our skin has to be able to live in these kinds of conditions and tolerate them. And then we also have special phagocytic cells, white blood cells that can go around eating things, that are called skin-associated lymphoid tissue salt Langerhans cells. So not only do we have a barrier, but there are, sentries on the top of that wall, on the top of that barrier, going around and taking out anyone who tries to get in. Then we do have openings in our skin. Our eyes, our nose, our ears, our mouth, our urinary tract, our digestive tract on the opposite end. Those are all our mucous membranes. Mucus because they actively generate mucus. They're wet. And mucus is a really nice food product for a lot of bacteria. But mucus is very, very sticky. So these mucous membranes trap pathogens. The bacteria get in through your eyes, through your nose, through your mouth, and they stick there because you got all this sticky mucus there. And then what we will end up doing is either we will lose that mucus out or in. And one example is in your respiratory tract. To prevent things from getting into your lungs are a very, very sterile environment. To prevent things from getting there, we have what's called the ciliary elevator. So you have all these cilia in your respiratory tract that literally take the mucus that your body creates and all the bacteria that gets trapped in there, and they brush it up, and then it gets to the back of your throat and you feel a little tickle and you swallow it, and then you digest your mucus and all the bacteria that came up in it, or you cough and spit it out. So it's clearing actively all of the mucus and trapped debris from your airway. And we have not necessarily the same ciliary elevator, but we have similar mechanisms. We tend to flush out the mucus whenever possible to get the bacteria that's stuck in there out. We also have immune cells that tend to live in our mucous membranes, that their job is to kind of seek out invaders and destroy them if they are able to get out of that trap. And then one thing that we're going to hit on a lot today too are these pathogen-associated molecular patterns, or PAMPs. What does that mean? These are basically the antigens that the cell is looking for. So they are conserved areas, meaning these particular structures tend to be ones that the cell needs to keep the same. They don't allow it to mutate very often. So they're conserved, they're not us, and they're easily recognizable. Something like bacterial flagella. Those are different than eukaryotic flagella. So we can immediately say this thing is not us because it's not made the way we would make our flagella. Or peptidoglycan, that is only in bacteria. So in that case, if I see peptidoglycan, there's a foreign invader around because that's not something my body makes. Things that are just associated with pathogens, that's what we're talking about here. Any questions about this aspect of the innate immune system? Okay, so we're going to get into one more aspect of the innate immune system, and that's our phagocytic cells, our white blood cells. So if you don't know, blood really has three components. It has plasma, which is where all of the nutrients and wastes get dissolved. It's the water-like component of the blood. And then we have red blood cells. They're called erythrocytes. The word cyte means cell, erythro means red. And then we have white blood cells, or leukocytes. That just literally means white cell. And then there are platelets. Platelets are not cells. They are basically cell products, we call them. They're like little proteins that go around and help you clot your blood. And that's important because a clot is a barrier. If your skin accidentally gets cut open, that's a breach in the defenses. So a clot will come in and reform that breach, put a barrier there to prevent things from being able to get in. So these platelets are technically a part of the immune system because they're restoring our physical barrier to prevent things from coming in. But we're going to spend most of our time here with the white blood cells. Red blood cells, they have no immune role. Their job is to carry oxygen around, and that's basically it. And in case you don't know, red blood cells don't have a nucleus. We remove the nucleus after they're made, and we pack them full of hemoglobin to hold as much oxygen as possible. And so they aren't able to take care of themselves and fix themselves. So our red blood cells have a definitive lifespan, and we're constantly making new ones from our bone marrow and then destroying them in our spleen and then making them and destroying them. And it's a whole process. But with the white blood cells, they do a lot of cool things. The two classes that we're going to get into are the myeloid cells and the lymphoid cells. These are just different types of white blood cells, different categories. So the myeloid cells, think of these as the phagocytes. Phago means to eat or food, so this is the cell that eats. What these guys do is they go around finding foreign invaders and they literally swallow them, eat them, and destroy them. So that's the myeloid cells. The lymphoid cells, these are ones that are going to be part of our adaptive immune system, our T cells and B cells. They're the ones that help with memory. They're the ones that help us target our cells that have already been infected, things like that. So in the myeloid cells, you're going to have things like neutrophils and monocytes. These are our primary phagocytic cells. Monocytes can become what we call macrophages or dendritic cells. Those are forms they can kind of evolve into. And then basophils and eosinophils are also similar in that they are phagocytic cells, but they have different roles. Eosinophils tend to be more in response to things like allergens and eukaryotic invaders, particularly parasites. So we see really high eosinophil counts if you have a high parasite load or if you are having really bad allergies. Then we're going to get into these two cells a little bit more later, but T cells, their job is to monitor our cells, basically. They watch our cells, and if they're behaving poorly, they attack our cells, or they can help stimulate the immune system. B cells, their main job is to make antibodies. So that is really why we have B cells. Technically, they are also phagocytic, but that's not their main job. They eat other cells to create antibodies. So that's why we don't really categorize them as purely a phagocytic cell. They can eat other things, but only because they're trying to sample, okay, what antigens do you have so I can create an antibody for it. And then these are just interesting pictures. Our white blood cells have really fascinating appearances, and you can actually tell what they are just through looking at them. If you've ever had to do a blood differential, you have to literally sit there and look at someone's blood and count how many neutrophils, how many eosinophils, how many lymphocytes and monocytes. It's very tedious. But they all have different lobular nuclei, and they have special granules inside that we're going to talk about later that facilitate their ability to kill the bacteria that they take in. So any questions about these guys? Have you guys heard a lot of this before? I'm seeing a lot of like, oh, okay, I've heard this. I'm seeing bored faces, so I guess that's better than panic. All right, so let's talk about these in a little bit more detail. The monocytes, they phagocytize foreign material, and then they can differentiate into macrophages. These are the ones that are really the first line invader, or the first line in our defense against the invaders after our skin. They're the ones that are patrolling all the time, that as soon as something sneaks in, they find it first. Dendritic cells are interesting. If you know the term dendrite, where do you normally think, what cells do you normally think of when you hear the term dendrite? Yeah, the dendrites are the kind of like hair-like protrusions that come off of the cell body. And so these cells have those as well. You can see all of those protrusions here. And their job is, once they take something in, they become what we call a professional antigen presenting cell. So once they swallow it, these macrophages, they're just kind of like, let's clear these guys up and kill all these guys. The dendritic cells are like, okay, I'm going to eat this, and then I'm going to put little pieces of its body outside on my surface so that the rest of the immune system can sample all these different antigens and respond to them. So it's like taking the heads of the enemies that are trying to invade you and putting them on pikes on the wall, literally. And then people can be like, oh yeah, that was George. Now we know, go attack George's family. That kind of idea. So these are antigen presenting cells. Technically, both of them do this, but dendritic cells, this becomes their primary purpose, whereas macrophages can continue to go around swallowing other cells later. So they take the antigens that they eat and present them on their surface. Really gruesome when you think about it. Our immune system is cutthroat. All right, so we're going to talk all about how this phagocytosis happens. So first is contact. The macrophages have to sense that there is a foreign invader, and they won't take that foreign invader up unless they recognize a specific PAMP, a pathogen-associated molecular pattern. So literally they have receptors on their surface of the macrophage that have to interact with one of these PAMPs and say, oh, I recognize this. This is peptidoglycan. They bind together, and then the macrophage will actually take its cell membrane and engulf the bacteria. So it doesn't pull it in the way some cells do with endocytosis. It actually pushes out its membrane to surround the bacteria and kind of like making those pseudopods. This creates the bacteria. This does not create the bacteria. What it does is it creates a vesicle that the bacteria is now stuck inside. And we call that vesicle the phagosome. So we have the bacteria stuck inside a little phagosome now. And the bacteria is fine in there at this point. Nothing bad has happened to it yet. So some bacteria evolved for this particular lifestyle. They want to be internalized by a white blood cell, and they have found a way to kind of defeat all the defenses of the white blood cell and live inside the phagosome. And they kind of go in there and they stay undetected. So that's not a good thing. But Normally, what happens is at this point, a lysosome will fuse with the phagosome. And if you don't remember, a lysosome is like the stomach of the cell. It has a bunch of digestive enzymes. Sometimes it can have a really low pH. I don't like calling them lysosomes because they're technically something different, but there are also structures that we deem peroxisomes, that their job is to create a lot of hydrogen peroxide to kill things with. We're kind of using the term lysosome broadly to cover all of these types of vesicles right now. So digestive vesicles will fuse with the phagosome and create something that's called the phagolysosome. This is basically like a full stomach now. We've put the food in the stomach, the digestive enzymes are in there, and we are going to start breaking down that bacteria. How does it do that? It does it with enzymes that actually do break down food through creating reactive oxygen species, which we'll talk about more in a little bit. Sometimes these lysosomes will also create a very low pH environment to destroy the cell. And in the end, we get a bunch of digested cell products in the phagolysosome. Make sense? Have most of you heard this before? What? This one, yeah? Okay. That's what I thought. I feel like this stuff, most of it is stuff you've heard before. All right, so the bacteria dies. And we have all this detritus, detritus. I've heard it both ways. I say detritus, sorry if you don't like that. This is antigens now. This is antigen soup. We've destroyed this bacteria. We have all these proteins and carbohydrates that are just from the bacteria. We don't necessarily want to lose this stuff before we create a response to it. We want to remember these pieces for the future. So in the end, we end up sampling these in many different ways. And these can be the things that then go on the surface and get displayed with our antigen presenting cells. And then we can also release them into the environment for other cells to sample and remember in the future. So just keep that in mind, that this is waste product, but it's waste product with some important information. So we got to look through that person's trash before we let it go to the dump. Any questions? Cool. All right. So just, I love pictures of this happening or videos of this happening because white blood cells and red blood cells are pretty big. And you can see them hunting down the bacteria. There's little bacteria right there being hunted down and then eaten by the white blood cell. It's just a lot of fun. Crazy to think this is happening inside your body right now. And it took a bunch of bacteria all at once. So it doesn't necessarily just have to be one bacteria at a time. It can take a bunch of them all at once as well. Because if bacteria are dividing, they often will divide and stay somewhat connected or close to each other at first. So if you can only swallow one at a time, that's a problem. You probably want to take as many as you can. All righty. Now, as I mentioned at the beginning, there has to be a connection. There has to be a moment of contact and recognition. So for all phagocytes, in order for them to proceed, they have to recognize the surface antigens and be able to take them up that way. So we've got all these receptors on the surface that can interact with a bunch of different antigens. And once that connection happens, then the whole process will begin. So one way that we recognize all this is through something called opsonization. Weird word that basically means we coat the bacteria in materials that make it easier to find and swallow. So one way that we do this is we coat it in antibodies. So if we cover a cell with antibodies, it can't move around as easily. It's much more sluggish. It's much more likely to get found. And then once it gets found, the constant region of our antibody gets recognized by our our phagocytic cell and more easily taken up. So just one way that we help make our job easier is opsonization. We're not going to get too much deeper into that. That's about it. Any questions? All right. So let's talk about how the phagolysosome kills the bacteria. There's two different ways: the oxygen independent and the oxygen dependent killing pathways. So oxygen independent really relies on enzymes. The lysosome will have a bunch of different enzymes in it that degrade biomolecules. So we want to break down the lipids, the proteins, all that stuff within the bacteria. So proteases, glycosylases, all that. But there's three very specific ones that help us against bacteria. Lysozyme is a really interesting one. This enzyme specifically degrades peptidoglycan. The way I remember this is zyme is kind of like an alternative enzyme ending instead of A's, and lyse, we're breaking the cell. So if I target the cell wall, remember from way back in the beginning, if the cell wall gets broken down, the bacteria is more likely to lyse due to all the pressure of water coming in and creating that turgor pressure. So it can't fight it, and the cell is most likely to lyse. So that's how it works. It targets the peptidoglycan. Very cool thing about lysozyme, it's not just in our white blood cells, it's in your tears and it's in your saliva. So you got it all in your face all the time, because these are heavy entry points for bacteria. So if we can kill them in our eyes and our mouth before they even get into where our mucus is, the better. Then we have lactoferrin. This one disrupts iron metabolism. A lot of times bacteria are coming to us because we are bags of nutrients for them. We are a nice warm place that has a perfect pH, that has a stable temperature, that has lots of water and lots of iron. In all of our blood, we have tons of iron in there. So iron is crucial for pretty much all organisms to live. And if you can get the iron, you kind of win. So they want to steal our iron from us. And if we prevent them from being able to use it, they're going to die now. And then we have defensins. These are small peptides that are embedded in the membrane. So what they do is they go into the bacteria, they embed in the membrane, and what they do is disrupt proton motive force. They basically make it so all of the ion gradients that the cell has created, they bring them to equilibrium. So the cell can't power any of its normal uptake mechanisms or its flagella or anything like that. Sucks to be them. That one's cool. The oxygen independent pathway is cool, but for some reason I find the next one much more interesting. And remember that oxygen is very, very deadly to all life, not just bacteria, but to us as well. So we want to detoxify reactive oxygen species with superoxide dismutase or catalase whenever possible. But we can also create on purpose reactive oxygen species to kill things for us. And that's exactly what these cells will do. They have enzymes called NADPH oxidases, and they are going to make ROS. They're going to make superoxide, the worst kind of ROS. And if that one doesn't work, we can also convert it to hydrogen peroxide, which will still kill cells just as well. So we create all of these reactive oxygen species or reactive nitrogen intermediates. They're not quite as good at destruction as oxygen, but they're close enough. So we'll create one or more or a mix of all of these guys. And we call this mechanism the oxidative burst. So we put them into the phagolysosome, and then we just hit them with all the oxygen we can, and the cell can't handle that much stress. It tries to mitigate the stress as best it can, but it's so much, and it can't escape the phagolysosome, so they die. Really crazy. Any questions about that? So cells that can produce the enzyme catalase to take that hydrogen peroxide and turn it into water and oxygen gas, they're going to survive this a lot better than cells that can't produce catalase. So we end up calling catalase a virulence factor. It's something that the cell, the bacteria has that makes it better able to survive and more able to attack our cells and survive in our cells. So we don't normally think of catalase as a virulence factor. You just think, oh, it just helps them survive in oxygen. But when we put it into this context, it can be a virulence factor. Any questions about that? All right, done with innate. Let's talk about adaptive. T cells and B cells, that's adaptive immunity. So with this one, we've just engulfed and destroyed invaders, but the adaptive immune system asks, okay, we took care of them, but what if they come back? So they want to take that detritus that was released, sample it, and remember it for the future. That way they can respond much faster in the future. It's saying we need to remember how these guys came in and attacked us, so we shore up those defenses a little bit more. This one is all about specificity. The innate immune system kind of kills whatever it comes into contact with indiscriminately. This one is targeted. It's going for the exact invader that it knows how to destroy. All right, ready to do it? Let's go in. There's two subdivisions of adaptive immunity, and that's humoral and cell-mediated. Humoral is basically B cells, and cell-mediated is T cells. And that'll make more sense as we explain their jobs a little bit more. So the reason why it's called humoral immunity is it's relating to the humors of the body, which we deemed all of the liquids in the body to be the different humors. So antibodies live in our blood, and so they are part of the liquid portion, the plasma portion of our blood humor. Whereas cell-mediated immunity, we're not really dealing with a humor or a liquid in the body here. We're dealing with our actual cells. T cells go around monitoring all of our cells for signs that we've been invaded and taken over. B cells are going to be in the blood just patrolling with their antibodies that they create. So antibodies are just proteins, they're not cells, and they specifically target antigens. They can bind to an antigen. And when they do, they make it easier for our cell to then attack that particular cell that they've bound to through things like opsonization. Are we ready to get into these cells a little bit more? Let's do it. So let's talk about the structure of antibodies first. They're technically called immunoglobulins or IGs, but antibody is a much easier word to say, so we tend to say that. But when you look at them, they all have a name that starts with IG. That's why I'm bringing their full name up. So we make them in response to an antigen. Every single antibody is specific to a particular antigen. It can't bind to any antigen it wants. It can only bind to one particular thing. And they are just floating through our blood. They circulate through our whole body, and they will randomly bump into things. And if it happens to be an antigen that they recognize, they will bind and stick to each other. They are made by B cells and plasma cells. Plasma cells are just superpowered B cells, basically. B cells are kind of a precursor cell, and they can differentiate into plasma cells. A plasma cell, its entire job is to just crank out as many antibodies as it can. B cells can also differentiate into memory B cells that, kind of wait. If we see that disease again, then they'll make plasma cells right away, and those plasma cells will then flood the body with all the antibodies that we need. So let's get into the different antibody classes that matter for us and the structure of an antibody. So they are proteins, and they're Y-shaped. This is the space-filling model here, but this is the more, structural model. And it's made up of 4 polypeptides, 2 light chains, and 2 heavy chains. And they are connected to each other with disulfide bonds. So technically, none of these is kind of free-floating. They're all connected to each other into one molecule that works together. They have 2 antigen binding sites at the top, made up of the heavy and the light chain together. And then at the bottom, they have the constant region. And this is one where our body will recognize and bind to that side. So the constant region is recognized by our immune cells. That's this part down here. And the variable regions are the antigen binding sites. So these ones go around and bind the invaders. This one then binds to our cells and kind of messages our cells and says, hey, I caught something. Take a look at this. We've heard this before too. Especially if you guys want to do medicine, I feel like you learn this stuff early if you're at all interested in a medical career. All right. So there are 5 different types of antibodies. We're only going to talk about 3 here. IgD and IgE, we're not really going to get into. So IgG, this is the most abundant antibody in the body. It's in all of our blood, all that fun stuff. It's going to be the one where when you go and get a blood test done to see if you've had a disease before, these are the antibodies we're looking for. They're the ones that circulate that say, yeah, you've had this before. They are involved in opsonization. They can cross the placenta. So these ones can go in and help give immunity to a baby. And they can activate complement, which is just a way for us to stimulate our immune system. Because I know we didn't really get into that. IgA is most often in this dimer form. So we're taking this normal antibody shape and we're kind of messing with it already. So it's most often found in this form here. It's not really found in the blood. Instead, you're going to find this one in the mucous membranes. So its job is to kind of sense and bind to things that are trying to get through our mucous membranes. It's found a lot in tears. It's found in things like breast milk, all that stuff. And they're interesting because they can control our gut microbiome. We're going to talk about that way at the end because that's something to think about. Those are bacteria too. Why don't we attack them? They live inside of us. Why does our body not seem to care about them? So it's partially because of these IgA antibodies. And then IgM, these ones form these really cool pentamer structures. So a normal IgG can bind twice to an antigen. The IgA can bond 4 times to an antigen, so to 4 different antigens. And this one is the highest, what we call valence. It can bond to 10 antigens. And this is going to be the first one that we detect in an immune response. So it's the one that we kind of like flood the body with as soon as we're having an immune response. Cool. All right. So let's talk about T cells now. T cells, they develop in the thymus. This is an organ that is much more active in childhood and adolescence than in adulthood. It seems to go into a kind of like senescence in adulthood, kind of indicating most of our immune growth happens when we're children and we're first being exposed to a lot of these things. But this is also one of those organs where we're still kind of like, why is it still there? We don't know. We're looking into it. And it's going to be divided, these T cells are going to be divided into 2 groups, helper T cells and cytotoxic T cells. So if we break down those words, we kind of get what they're going to do. Helper T cells are going to help the immune system respond to an infection. Cytotoxic T cells, these are T cells that are toxic to our cells, not bacteria cells. They're toxic to our cells. So they're going to go around and patrol and watch all of our cells and say, you're behaving badly. You've been taken over by a hijacker. I'm going to destroy you. We don't get into it here, but within this group of T cells are also T cells that can recognize our cells that have been turned cancerous. And they can actually destroy cancer cells early before they create a tumor and all that. All right, so the term lymphocyte just basically refers to a T cell or a B cell. So if you ever see that term, that's just what we're referring to. These are also known as killer T cells. And we're going to talk about binding of surface antigens and other. We're going to talk about a lot of binding. So we need to talk about surface proteins and how they bind. So in this case, the surface proteins of these cells are important. The helper T cells will have CD4 plus surface antigens. I'm sorry, my brain now is, we're getting to the end and it's hot in here. I'm fading fast. And then we have CD8 plus on our cytotoxic T cells, and we'll talk about those more in a little bit. Okay. So they come into play with this idea of the major histocompatibility complex, the MHC. This is our way of communicating, hey, I found an invader, this is who the invader is, or hey, I've been invaded and I need help. The MHC is technically a set of genes that's going to code for a specific set of surface proteins. But when we talk about the MHC, we're usually just referring to the surface proteins that it creates. And these are going to be antigen presenting proteins. They're going to present peptide fragments, so little pieces of protein that the T cells are going to be able to recognize through their CD4 plus or CD8 plus receptors. So when the MHC proteins are bound to these peptide fragments, we call it the MHC peptide complex. So this is actually one of the main ways that the cell says, you belong to me, and this cell over here does not belong to me. Self and non-self differentiation is really because of this. So we can even present self material and kind of tell the immune system, this is self, get used to it, don't attack this. Sometimes the immune system gets a little worked up and uppity and says, no, 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 that's bad too. I'm going to start attacking that too. And then you get things like autoimmune disorders. All right, there's two classes of these MHC receptors, class 1 and class 2. Class 1 is found on all nucleated cells. So these are on every single cell in your body except for red blood cells because they don't have a nucleus, and not on platelets because those technically aren't cells. And then class 2 MHCs, these are found only on our professional antigen presenting cells. So these are the ones with phagocytic ability. So any cell can present antigens, self antigens or not, but the class 2 are the ones that can also say, I found this stuff here, look at this, this is non-self attack kind of thing. So MHC class 1 is going to present peptides intracellularly from inside the cell. It says, okay, I found this in the cytoplasm and I'm going to bring it out. And this is going to be recognized by our cytotoxic T cells. The cytotoxic T cells are the ones that recognize our cells have been invaded and they need to be destroyed, either by a virus or a bacteria or whatever it is. So it makes sense that we're sampling from inside the cell to put on the surface because we're saying something is inside me that doesn't belong there. Here's a piece of it. Help me. I'm being invaded. I need to be destroyed. So that's why the cytotoxic T cells can recognize class 1. And for class 2, these are going to be recognized by helper T cells. These are the phagocytes saying, hey, I just killed this guy. Helper cells, we've got an invader. Mobilize the rest of the army, mobilize the rest of the immune system because we've got a fight now. Let's go fight. So the helper T cells will then go and activate our B cells. They can often activate cytotoxic T cells as well, and it just mobilizes our whole defense. Make sense? Okay. So just reiterating, MHC class 1 is saying these are intracellular antigens. Something from inside is being presented out. Whereas MHC class 2 is saying, I found this outside of us, I ate it, and now here you go. Those are the big difference there. I feel like I'm just repeating myself a couple times here. Much easier than immunology level difficulty, right? All right. T cells have receptors too. And we call these TCRs, T cell receptors. And they are going to recognize and bind to the antigens. So these are specific to antigens, meaning one T cell receptor only binds one antigen. So one piece of peptidoglycan or one piece of lipid A of LPS, something like that. And these are going to associate with the antigens that have been presented from the major histocompatibility complex. So it's going to say, okay, you've been presented, I'm going to now associate with you and create a response, either the cytotoxic response or the helper response. And this whole process is part of kind of like that signal transduction cascade, because we're going to bind here, it's going to cause conformational changes here that cause a signal to be sent, and the cell gets mobilized that way. And really what we're triggering is make more T cells. We've got a job now, let's go do the job, let's make more cells to go do the job. This is about as deep as we're getting with these guys. Alrighty. So on the cytotoxic T cells, these are labeled as CD8+. And so they're going to only be able to bind to an antigen that is attached to an MHC class I protein. If you're a T helper cell, you have a special TCR that is labeled CD4+, and you're only going to be able to bind to antigens that are attached to the MHC class II. Just reiterating that point, that's probably an important one for you to understand for the exam. Understand the difference between class I and class II of the MHC and what types of T cells bond to them and why. Okay. Now, once we have activated a cytotoxic T cell, what's going to happen is that cytotoxic T cells like, okay, I've been activated, that means this cell has a problem with it, I have to kill this cell now. So how do they do this? They first secrete perforin. Perforin sounds like perforation, we're poking holes in things, and we're going to actually literally poke holes in our infected cell. So our cell has a virus in it, we've sent out the signal for help, the help comes and says, you got to be destroyed now. We're not even going to take any chances. We're just going to nuke the town to prevent any bad guys from getting out. So they poke holes in it, disrupt the membrane integrity, and then they secrete granzymes. These are going to enter the cell through those pores that the perforin created, and that's going to trigger apoptosis in the cell. So we destabilize the membrane, make a bunch of holes in it, then secrete granzymes in through those holes and trigger the cell to kill itself. That's what our cytotoxic T cells do. And they do this viral infections, intracellular bacterial infections, like I said, cancer cells, all that stuff. Any questions? I'm trying to explain all this in like as simple a way as possible and not get caught up in a lot of the details here, because I feel like our brains are dead at this point of the semester. All right. So now the humoral immune response, we're going back to our B cells here. The T helper cells, they bind to antigens on MHC class II. And what this does is it produces cytokines, and cytokines recruit white blood cells. They're kind of like the rallying call for those guys. And so macrophages become better at killing when the helper T cells release these cytokines. And they also cause our B cells to differentiate. We make memory B cells and we make plasma B cells, plasma cells, and those produce a lot of antibodies. These B cells and plasma cells also have specific receptors like the T cell receptors, and they're going to be bound and then trigger the differentiation. So we don't want to differentiate if there's no reason to. We don't need plasma cells unless we know an invader is present. We don't want to waste a lot of this energy to make antibodies. Antibodies cost a lot of energy to create. So we don't want to make them unless we have to. So only when the B cells bind to an antigen are we going to say, okay, now create this guy that's going to make all of our antibodies. Make sense? Just all energy. Okay, this picture sucks. I'm sorry. It came from your book. There are much better pictures out there if you don't understand this one. But let's break it down simply. this is how we're going to activate antibody production with our B cells. So we start in the lymph nodes, where there's going to be antigens because they get into our lymph nodes. We've got invaders, they're inside, and most things end up in the lymph nodes eventually. It's a big rallying point. There's also lots of B cells in the lymph nodes. The B cells can recognize those antigens because B cells have antibodies sticking out of them. So the constant region is embedded on the membrane. They actually have certain types of modified antibodies that have a lipid-soluble part on their constant region, so they can embed in the membrane itself. And so these are now out there looking for antigens, and if they accidentally bind to one, the B cell is going to initiate a response. So this binding is all random. They're just all floating in the lymph nodes, and this B cell happens to have the correct antibody that matches that antigen that it randomly bumped into. And then when that happens, when the encounter occurs, the B cell is going to internalize what it just attached to. What we mean by that is it's actually going to do phagocytosis. It's going to say, hey, something bound here, let's pull this in and sample it and figure out what we need to do with it. So it internalizes it. The B cell will then process it and display it on its MHC class II. It says, okay, I've taken this in, it is a foreign invader, let's process it properly and stick it back out on our surface for the immune system to recognize and our T cells to formulate a response. So that's the first step of this. So T helper cells come along and they're like, ooh, there's something here on this MHC class II. There's an antigen for me to see. When that happens and the T cell binds to that MHC class II and the antigen in it, that binding is going to say, okay, B cell, proliferate, make lots of copies of yourself because you found something that's attacking us right now. So we need more of whatever antibody you know how to create to go and attack and bind to this foreign invader. So part of that is the B cells are going to copy themselves, and some of those copies differentiate into plasma cells. The plasma cells then pump out as many antibodies as they can. Those bind to the invaders and make them easier for our white blood cells to find and attack. We also make memory B cells, which are always going to circulate. And that way, when a second infection comes along, those memory B cells can immediately differentiate and make tons of plasma cells and respond right away to the infection. Normally, this whole process of taking it in, internalizing it, learning, making the plasma cells the first time is what takes around 2 weeks. But the memory B cells can differentiate into plasma cells and create a response within days instead of weeks. Any questions about that? Okay. So, coming back to this last point here, we have a microbiome. There's bacteria living on us and in us. That's another interesting point too. A lot of times we think that our gut is inside of us, and it's technically not. We are a donut. We have a hole that goes from one end to the other, and that is open to the outside. We're constantly taking the outside and putting it in. As a result, we can't really consider that part of our body inside. It's still outside, like our skin, just a little wetter and grosser. The inside is things like your lungs, your body cavity surrounding your intestines, surrounding all of your important organs, your muscles, your blood. Those are all inside your body. Those are the really, really bad places to get bacteria. We do not have a microbiome there. If the microbiome bacteria gets into those areas, usually we kill it. And we say, you are no longer nice anymore. You've stepped your bounds. It's like the friend who you suddenly find in your living room one day who's like, oh yeah, I'm moving in. No. You're my friend, but get the hell out. So we don't normally want to attack our microbiome, but we need to recognize you aren't self, and I can attack you if you overstep your bounds. So we have some really interesting ways to do that. We're not really going to get into it, but I know a lot of you guys in this class are super curious, so I thought I would discuss a little bit about it. The main way that we don't attack our microbiome is we have those firm boundaries. We say, you stay there and I stay here. You stay in my gut, you stay on my skin, and as long as you don't come in, you're fine. So that barrier is the first and main way that we don't attack our microbiome. The second one, we actually have special regulatory T cells, and they're going to actually our whole life be sampling our microbiome and kind of make our immune system say, hey, these guys are kind of here all the time. They're not doing anything bad. Let's ignore them. So we know that they're there, but don't respond to them. Like, don't have an immune response. Don't cause inflammation. Just stay calm. We also have the commensal bacteria, the ones that live on us and in us. They technically often will have modified PAMPs. So they won't have ones that are truly virulent and pathogen inducing that scare our immune system. They kind of have a modified version of it that our body doesn't respond to quite as strongly. So it's basically suppressing our immune system is a lot of the name of the game with this. We're going to say, okay, yeah, that's not self, but calm down. This one's okay. And then we also have specialized IgA antibodies that are going to constantly be binding to our commensal bacteria. And them binding says, it's okay, don't respond. We know he's here, we're watching him. So it's a lot of balance between we see you and we're going to calm down and we're not going to attack you unless you cross that barrier. Kind of weird to think about. I know it's a lot of personalization or personification of this stuff, but I feel like it makes it easier to understand. All right. Any questions? Cool. That was fun, right? A lot more fun than genetics, in my opinion. All right. So I will post your homework probably tomorrow. That's my guess. And then the next two classes, we get to talk about how bacteria attack and get around our immune system, and antibiotics and how they kill. All right. Have a good day, guys.